Dr. Sage here. In this set of video lectures, we're going to discuss the host defenses. Of note, this is just a cursory overview. For a much more detailed explanation, take an anatomy and physiology course. In particular, in this video, we're going to discuss specific immunity, which is the third line of defense. By the end of this video, you should be able to describe how the third line of defense is different from the other host defense mechanisms, list the 4 stages of a specific immune response, discuss 4 major functions of immune system markers, define the role of the major histocompatibility complex, and list 3 classes of MHC genes, compare and contrast the process of antigen recognition in B cells and T cells. Acquired specific immunity is a product of B and T lymphocytes. Lymphocytes undergo a selective process that specializes them for reacting to only one specific antigen or immunogen. Immunocompetence is the ability of the body to react with countless foreign substances. Antigens or immunogens are chemical substances that stimulate a response by T and B cells. They can be protein or polysaccharide molecules on or inside cells and viruses. Any exposed or released protein or polysaccharide is potentially an antigen. Usually, our own antigens do not evoke a response from our own immune system. Specificity refers to the fact that specific immunity is highly specific to the antigen against which the third line of defense is directed. Memory refers to the rapid mobilization of lymphocytes that have been programmed to recall their first engagement with the invader and rush to attack again. The principal stages of immunologic development and interaction include lymphocyte development and differentiation, presentation of antigens, antigen challenge by B and T lymphocytes, B lymphocyte response, which includes production and activities of antibodies, and T lymphocyte response, which includes cell-mediated immunity. Markers on cell surfaces are involved in recognition of self and non-self. Major functions of immune system markers include attachment to non-self or foreign antigens, binding to cell surface receptors that indicate self, MHC molecules, receiving and transmitting chemical messages to coordinate the response, and aiding in cellular development. There are 3 classes of MHC genes. The class 1 MHC genes, the markers appear on all nucleated cells, display unique characteristics of self, allow for recognition of self and the regulation of immune reactions, and each human inherits a particular combination of class 1 MHC genes. Class 2 MHC genes code for immune regulatory markers found on macrophages, dendritic cells, and B cells. They're involved in presenting antigens to T cells during cooperative immune reactions, whereas class 3 MHC genes encode proteins involved with the complement system. Cluster of differentiation are markers important in immunity. They're found on membranes of a variety of different cells involved in the immune response, and close to 400 have been described. Tissue macrophages ingest the pathogens and induce an inflammatory response in the tissue if appropriate. Tissue dendritic cells ingest the antigen, migrate to the nearest lymphoid organ, process and present antigen to T lymphocytes. Antigen-presenting cells include dendritic cells, macrophages, and B cells. B cells and T cells proliferate and differentiate when challenged by antigen. A clone is a proliferation of a particular lymphocyte. These are genetically identical cells, some of which are memory cells. The 3 main functional types of T cells include helper T cells, which activate macrophages, assist B cell processes, and help activate cytotoxic T cells. Regulatory T cells, which control the T cell response, and cytotoxic T cells, which lead to the destruction of infected host cells and other foreign cells. When activated by antigen, a B cell divides, giving rise to plasma cells. Each plasma cell has the same reactive profile. They release antibodies into the tissue and blood, and the antibodies attach to the antigen for which they are specific and mark it for destruction or neutralization. This has been your brief overview of the third line of defense. The rest of the videos in this series will go into more detail about the third line of defense. Until next time, this has been Dr. Sage.